Hi, welcome to AP Biology, timed AP Practice number 1 review. Hi, my name is Gail Pfizer. I'm from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. And yesterday, I actually enjoyed proctoring you yesterday. Watching the clock and being nervous with you, I felt like I had a big part in what you were doing. First, today's experience and yesterday was experience was designed to be practice with procedures as much as content and skills. And we have a really short agenda. We're just going to unpack this FRQ, and then I'm going to direct you to this video that I want you to watch. It's 5 steps to prepare for AP. So let's start with the question 1. And I had my students write these FRQ answers also. And this morning, I got some good feedback that doing through this experience, they watched the clock. They said that they felt better about things, that it seemed doable. And I hope you feel that way too. So I'm just going to read the first part, and then we'll break it apart into each little part from there. Okay, so Cryptosporidium Parvum is a single-celled eukaryotic parasite that infects human cells in the digestive system and causes illness. Although it is a eukaryote, C. parvum does not have functional mitochondria and generates ATP only through glycolysis. C. parvum uses the enzyme lactate dehydrogenase to perform fermentation after glycolysis. 2 chemicals, gossypol and FX11, are non-competitive inhibitors of lactate dehydrogenase. Researchers investigated the effectiveness of gossypol and FX11 as drugs to kill C. parvum. In the experiment, human cells were treated with different concentrations of either gossypol or FX11 after infection with C. parvum, and the relative growth of C. parvum compared with that of control cells was measured. And then we have figure 1 and figure 2. We have the effect of the different concentrations of gossypol on the C. parvum growth. And we have in micromoles, and we have relative growth of C. parvum in percent on our y-axis, okay? And then we have FX11 and the same X and Y. All right, so now let's look at each part. Okay, so A starts off with describe. And so remember, describe means provide relevant characteristics of a specific topic. So describe how C. parvum obtains the glucose it needs for glycolysis after it has infected another cell. And then the answer is C. parvum. Hopefully you have this in your answer, okay? C. parvum absorbs from its environment, which in this case takes glucose away from its host. So let me just briefly go over what's happening here. So you have, here is our host cell, okay? And let's draw a few parts because we have plenty of time today. Here's our nucleus, and I'm gonna go ahead and just put a mitochondria in here, okay? Because we're gonna talk about it later. And then we have our C. parvum, okay? Our C. parvum comes in and it infects the cell. So it means it's inside of the cell. And now it is, let's see, let's go yellow, well, maybe yellow. You can't see that. How about that? So now we have glucose in the cytoplasm of the cell, which would be going through glycolysis, right? For the cell, but the C. parvum is going to take the glucose from the cell, from the host cell, and use it. So that's where we're going to start. C. parvum absorbs from its environment, which is inside of this cell, and takes the glucose away from its host. All right. All right, so B, explain. So let's go look at explain, how or why, okay? So not just a description, but how or why is this happening? So explain the role of lactate dehydrogenase in enabling C. parvum to continue producing ATP by glycolysis. So hopefully your response included lactate dehydrogenase allows fermentation to convert NADH to NAD+, which is needed for glycolysis to continue making ATP. So let's look at glycolysis here. So here here's our glucose coming into glycolysis. We're able to make 2 ATP, And then along that process, we also reduced NAD plus and that's NADH. So now we have 2 NADHs and we have pyruvate. Well, to keep this going, we have to make NAD plus available again. So those electrons then have to get back off of, with the NADH has to be oxidized in order to be able to go back to glycolysis to make more ATP. So then the lactate, that's where the electrons are going to go. So it was pyruvate, and now we're going to get those electrons back and we're going to be lactate. So this enzyme is going to allow for the fermentation to convert NADH to NAD+, which means, let me get something else. So this was our enzyme. Say this is our enzyme, and this is our lactate dehydrogenase, okay? And so if this was to be our substrate that we were putting, that fits into the enzyme, then our product would be NAD+, okay? So this is what we're using this enzyme for. It is for NADH to become NAD+, okay? And we need the NAD+ to go back into the cycle of glycolysis so it can make those 2 ATP. All right, so C is identify, and identify is pretty easy. Sometimes you can do it with just one word will answer the question. So identify the independent variable used in the experiment. And I hope your response included the concentration of the chemical is the independent variable. So they did not compare FX11 on the same graph here, okay? So we're talking about concentration. So those are somewhat 2 variables, okay, to compare later from each other, but they're going for the same goal. So it was the concentration of each was the actual independent variable, okay? So it changes in each of them. That's what they were changing, the researcher. All right, so here we have identify again. Identify the difference between the control cells and the experimental cells used in the experiment. And I hope your response included an identification of the characteristics of the control cells as the same types of cells infected with a parasite but not treated with any chemicals. So let's do this. Here is experimental cell, here is control cell, and these are human cells, okay? Those are human. And let's do all our parts because it's going to be meaningful in a little bit. That's a mitochondria, by the way. Okay, now they both have something else too. So these are 2 human cells, and now they have the C. parvum. Now the control group is the human cell with the C. parvum, but the treatment group is, the treatment group is the human cell with the C. parvum with the, let's just make this their chemical, okay? That could be our glossy pole or FX11. So that's the difference in the 2. All right, justify the use of the control in this experiment. I know over the past 6 weeks, I've been harping on this. I hope that you included the control is used to determine any change in C. parvum's growth is due to the treatment. So let's look at what the word justify means. Provide evidence to support, qualify, or defend a claim, and or provide reasoning to explain how that evidence supports or qualifies the claim. So I always ask my students to provide evidence and some biological reasoning why this makes sense, okay? And in this case, when we talk about a control, we know our control is the one with no treatment, right? So we have to also, when we're talking about a control, say that we're using the control in order to be able to determine if any change that's happening that we're measuring is due to just the treatment and not just under normal circumstances. Okay, another justify. Justify the researchers using a different range of concentrations for FX11 than was used for gossypol. So I hope your response included FX11 had minimal impact on the growth of C. parvum until it reached a concentration of greater than 30 micromoles, which was the maximum concentration of gossypol that was tested. So we see at 30 here, with the gossypol that we have no growth, it's over. But at 30, look at 30 on FX11, it's still way up there. So it's going to take much more. So that's why they had to extend this, the concentrations and keep going. All right, based on the data in figure 1, identify the concentration of gossypol that reduced C. parvum growth to 50% of that in control cells. The concentrate, and I hope you included, and this was, I'll tell you this later, never mind. All right, so the concentration of gossypol that reduced C. parvum growth to 50% of that in control cells as 15 micromoles. So that would be this point right here. And then what I was going to tell you is that I have, my students took this exam and I've started grading them. And this seems to be the experimental design, they're doing fantastic on it. Everybody so far has gotten all experimental design questions correct. And this was a particular one that everybody got correct. All right, so researchers discovered a strain of C. parvum that expresses a functional variation of the lactate dehydrogenase gene. A DNA sequence comparison showed that the variant differs from the normal sequence in the region that codes for the enzyme's allosteric site. Predict the effect of the FX11 treatment on the C. parvum that express this variant of lactate dehydrogenase. And I hope you said that FX11 will have little to no effect. I'm going to explain the whole thing on the next slide. But predict, let's talk about predict. Predict or make a prediction. Predict the causes or changes of the effects of a change in or a disruption to. And we can guarantee that that's what in question number 2 on this exam and on the 18th, it's going to have this disruption or change of some kind, okay? One or more components in a relationship, pattern, process, or system. So now let's on the next answer, because it's going to tell us to provide reasoning to support our prediction. So this is kind of like justification. So the allosteric site is the region of the enzyme where a non-competitive inhibitor like FX11 would bind. It is likely that changing the sequence of this region would decrease inhibitor binding. So let's talk this through what's going on. So here is our enzyme, okay? So there is our lactase dehydrogenase. And here is our NADH, right? And we want this, we want that. So this inhibitor, these chemicals, then would do this. So here's our lactase, there's our enzyme, okay? And now they have an allosteric site. So let's draw a little allosteric site and then we'll get rid of a dot there. Let's do it again. Okay, then we have an allosteric binding, site binding. Now, when that happens, then what happens to our active site? Our active site changes, right? So now will our NADH fit? Okay, will this fit in there? No, it won't. Okay, so there's our NADH and it won't fit. So it can't go in there. So now if we can't have the NADH won't fit in the active site, then we can't make NAD plus, right? And so if we can't make NAD plus, then we can't make more ATP. Then if we can't make more ATP, then what's going to happen to our cells, to our C. parvum? They're going to die. Okay, so that's how the chemicals are working with that allosteric site. So now let's do what the question's actually asking. So now if we have, here's our enzyme, okay, and we have our NADH fits, and now we have a mutation or a change in the sequence, and now our allosteric site is different. Now our FX11 will not fit in that, will it? So what effect will then the FX have on this? Okay, so what was the question? Let me go back for a second. So predict the effect of the FX11 treatment on C. parvum that express this variant of lactase dehydrogenase. Okay, so what effect will then this F11 have on this NADH being broken down into NAD+. It will have none, right? And then everything I just said is to provide the reasoning why. So the allosteric site is the region of the enzyme where a non-competitive inhibitor like FX11 would bind. It is likely that changing that sequence would decrease inhibitor binding. Okay, so if you will, when you justify, if you just talk through what is happening and why it's happening, then you'll be sure to score that point. Okay, so now this one, explain how gossypol in FX11 might be used as drugs to treat C. parvum infection in humans without negatively affecting human cells. So the drugs were doing what? They had an allosteric site, and they were making the enzyme that needed to go from NADH to NAD, right? They were disrupting that enzyme. So to go back into glycolysis, but what happens with pyruvate in a human? Okay, so we have a human cell, and so this was the point of the mitochondria. There's our nucleus, there's our mitochondria. So we have our glucose molecule, and we're going to go through glycolysis, and then we become pyruvate, right? And then are we going to go through fermentation? No, so we don't need for this NADH to become NAD+ in that system. So we have our own, we have this glucose is then going to be broken down to pyruvate. The pyruvate is going to be go through pyruvate oxidation, become acetyl-CoA, go into the Krebs cycle, and all those things will happen. So this will, while the drugs will prevent C. parvum from generating the ATP it needs to survive, human cells have a mitochondria and do not rely on fermentation to produce ATP, so they are likely to not be harmed. And that's why we have the mitochondria. Okay. All right, so that was it for question 1. I hope you either learned something along the way, feel more comfortable about the test process itself, or feel better about what you do know, or have some idea of what you need to work on from that question. So now let's look at our question 2. We have a cross section of a chloroplast showing membranes, and the spaces between membranes is shown in figure 1, okay? And figure 1, a diagram of the cross section of a chloroplast. So we're going to break this into parts and then take a look at each one just like we did in question 1. All right, so A, describe the major process that takes place in this eukaryotic organelle. And your response should have included the major process that takes place in the organelle as light providing energy, okay? Energy is key, and this change in energy form to talk about when you're talking about what goes on in the chloroplast. So light providing the energy to fix carbon into organic molecules, okay? Because that's the point. That's the point of what's going on in this molecule. It is not to make oxygen for us. That's a byproduct. The point is to make a food source for the plant itself. So light's coming in, and there's going to be an energy change in energy form, and we're going to go from light energy to chemical energy by the time it's over, okay? And we're going to fix carbon in the meantime to do that. All right, so B, explain the function of the structure with an X in figure 1. So X is pointing to the outer membrane. The presence, and this is what you should have included, the presence of organelles allows the compartmentalization of specific structures to more efficiently carry out specific metabolic functions. That's one of the first things we talked about with organelles and having membranes was the compartmentalization. So to keep everything, so we're able to build up these hydrogen ion concentration gradients so we can make ATP, and so we can then run the Calvin cycle and all of that. So very easy compartmentalization to carry out. So these two words were key here, compartmentalization and efficient metabolic functions. All right, so C, identify the location where carbon fixation occurs on the figure below. And you could have just said, well, where does carbon fixation occur? It occurs in the stroma, right? And so this right here would be the stroma. So B is your answer for C. That was real simple. Okay. All right, and D, and now I'm going to give a disclaimer that I wrote C, D, A, E, and F. And so I have my idea what the answer should be. And there may be some variation after grading a few papers today of my own students, but I'll tell you what I've seen. Okay, explain the formation of a proton gradient in the production of ATP. And your response, what I was looking for, what I wanted you to know, what I wanted my students to know was, as electrons are moved through the electron transport chain, hydrogen ions, protons, are actively transported to form a hydrogen ion concentration gradient in the thylakoid lumen that will move from high to low concentration through ATP synthase, converting ATP and P to ATP. So let's go through that on the diagram. Here is, we have the splitting of water, we have electrons, we have light stimulating the chlorophyll molecules, electrons leave, they go on the electron transport chain on a carrier, and then the hydrogen ions are pumped through, and then we're building up a gradient. This is inside the thylakoid space. So if this was a continuation, this would be inside the thylakoid. So this hydrogen ion concentration builds, and then we have, it's going to go through, and then it's going to turn that enzyme, and then it opens up an active site for ADP and P and forms ATP. So this is what I want you to know about the production of ATP on the thylakoid membrane, okay? All right, and then E, predict the effect of the removal of the final electron acceptor on the rate of carbon fixation by the chloroplast. So here's that disruption, okay, that's going to be in question 2. And your response, hopefully, hold on just a second. Okay, your response hopefully included removal of the final electron acceptor would decrease the rate of carbon fixation by the chloroplast. So here's, let's get, what color could I use that you can see? Let's stay with green. All right, so here is where we're going to pick up electrons, right? And we're going to form NADPH. And then we're going to take, NADPH is going to be out in the stroma, okay? And then that's where the Calvin cycle is. And that's where the electrons will then, NADPH will be oxidized, and then that Calvin cycle will be able to produce organic molecules from the carbon dioxide, okay? Now, if we did not have that, there's our disruption, then, or didn't have NAD plus, then those electrons aren't getting off of there, right? And this has a standstill. And this is where some of my kids went, but they didn't head towards, but they did it in a way that they talked about this would shut down the production of the ATP, and the ATP wouldn't come. Both of them actually would be also acceptable. NADPH and ATP are not being produced to go to the stroma if this shut down. Okay. All right, and justify your prediction. The final electron acceptor, NADP plus, is reduced to NADPH. And okay, and then NADPH carries electrons to the Calvin cycle in the stroma to be used along with CO2 to form organic molecules. That's what I just talked about in the previous slide, was the justification. So predict what would happen. The removal would decrease the rate. I forgot all about saying what the answer was to that. Okay, so prediction, it's simple, decrease the rate, okay? And make sure you use words increase, decrease specifically, okay? And then the justification, I just already ran through that. All right, I wish that I could know if you had any questions at this point about either one of these questions, but I'm gonna leave you with this. I want you to let's see, I want you to watch this one right here, okay? It's 16 minutes long, but it has like the top 16 minutes of information to help you. You already are going into your AP classroom and you're working on your practice questions, right? And then you'll be back tomorrow for our next round, our practice set 2 with Ms. Kate, and she'll have a special guest star on Thursday, okay? And then you've been to the demo site, hopefully my students and I ran through that yesterday morning in my classroom, and today they actually did it while they were answering the question that of last night's questions. And so with not with the demo site, but actually the deciding which way they were going to go, were they going to split screen and all of that. There's your documents, your checklist, and let's see. I think start here, okay? Use your time wisely, start there, practice there, and get your list printed. That's my advice for right now, okay? I'll be back on Friday, and for our final send-off video, Ms. Kate will be here tomorrow for your second set of questions, and I'll be watching, and I'm going to write with you too from where I have not seen her questions. So I'm going to be right there with you, and I'm going to then share in your joy or something else after it's over. And then you'll have on Thursday, you'll go over her practice questions, special guest star, so make sure that you attend both days. And then we'll say goodbye on Friday. And that's it for today. Thank you, goodbye.